I'm going to talk a little bit about dosage calling, which might be something.
This meeting is being recorded.
Which might be something new to some folks who work with diploids only. And then I'm going to show some examples of linkage and QTL mapping we've been able to perform, and select some genes, convert them into Cas markers, and then that's the examples I'm going to show after. And in addition to the markers for assisted selection, we are going to discuss a little bit about the challenges of using Cas markers for quality assessment and quality control in sweet potato. All right. So dosage calling is an interesting concept. We don't call it dosage calling, we just call genotypes, right? Or genotype calling. And it consists of counting a certain allele, let's say the alternative allele, right? So this one has 0 Cs, 1 C, and 2 Cs, right? So 0, 1, 2 would be the dose of Cs, right? Or the alternate allele. This is an example of how we would see it in a diploid hypomia trif, the full seed population. It's a right. So you can see some of the individuals fixed for one of these SNPs, right, for one of the alleles, and some of them have that express the existence of two in mapping population. It's of course segregating 1-to-1 or 1-to-2-to-1 as expected, right? So this is nice. This is what everyone has seen so far for sure. But then things start to get a little trickier as we increase the diploid level. This would be an example for potato, for example. So now our dosage calling go from 1 through 4, right? And again, we are doing the same work, right, of counting the number of alternate alleles. And this would be like the way we would analyze the data, right? And I forgot to tell, but these are read counts of genotype by sequencing protocols, diverse protocols, right? So we are basically counting the number of reads that has that specific allele. So if the individual only has reads for one specific allele, we call it homozygous, right, or 0. If it has only reads for the other allele, we call it homozygous 4, right? But now instead of having only one heterozygous class, we have 3, right? Which of course gets things a little bit messier, but it's doable, right? We learned how to do it for tetraploid species for quite some time now. Now we face the case of sweet potatoes, right? Which is a hexaploid species. So we again increase the number of heterozygous classes, right? And as you can see, the challenge of clustering individuals into these 7 possible classes, right, is bigger because now we have individuals which are fixed for one allele or has one allele of the alternate allele and 5 alleles of the other allele and so on, right? But it's also something that we learned to deal with, right? It took us a little bit longer to figure the protocols and everything, but now we've performed this analysis for a couple of linkage mapping populations and some diverse populations, and that's what I'm going to show now. Name of the protocol we developed at the time within the Genomic Tools for Sweet Potato project was called GBS Poly. And at first we actually got over 1 million SNPs for dosage calling, which of course is not something that we need for linkage mapping, but it was just something that came out of the first protocol, right? So we ended up with 30K SNPs mapped into a fully integrated linkage map, meaning that we estimate jointly the recombination frequencies of individuals in the two parents at the same time, right? Instead of having two separate maps. And by doing so, we are able to estimate the composition of each one of the individuals, right? And know exactly which homologous chromosomes exchanged pieces during meiosis, right? Through recombination. This is just an example for one individual, right? You can actually trace it back to the parental haplotype. So this is very nice because, of course, by having that information or estimating the conditional probabilities of the individual being, homologous A, B, and C, and for example, G, H, and I for one specific location allows us to detect QTL, right? So we came up with a model that uses random effects to be able to deal with the many classes we have in hexaploid potatoes, right? We can have for one single locus up to 400 different genotypes. So this is, of course, something that cannot be dealt with fixed effects, right? So we used random effects and started using it for quantitative and more qualitative traits. This is just an example of the study of the genetic basis of negatively associated beta carotene and starch, right? Which is actually something that sweet potato breeders are interested in, of course, and they have been dealing with these negatively association for quite some time. What we found was that the same haplotypes that decreased the values of dry matter and starch in sweet potato, or at least in this population, was exactly the same haplotype that made beta carotene and flesh color increase, right? So a combination that it's not desirable for African communities, at least, right? Because they are more used to high dry matter and low beta carotene, but we wanted to increase beta carotene, but the fact is that they're actually we have actually linked genes for controlling each one of these pathways, right? Which make them negatively correlated. As one increases, the other decreases, right? These QTLs were located on chromosome 3 and 12. Another population, actually, the reciprocal, was used at NCSU for mapping nematode resistance. Now the GBS poly delivered over 115K SNPs. We again were able to build a linkage map with 15 linkage groups and almost 15K SNPs, right? And again, by using, and by the way, these are the haplotypes, right? The way we organize all those variants into 6 homologs per parent, right? So by using that genetic map again and the methodology we have developed for QTL mapping, we were able to discover that there is a major QTL on chromosome 7 controlling the variation for nematode resistance, right? This is for incognita, myeloidogenic incognita. And that 2 haplotypes were the ones responsible for decreasing the number of root counts, root knot counts, right? So of course we were like, all right, so now we know which haplotypes where the QTLs are, right? And which haplotypes we need to target for performing marker-assisted selection. So let's try to do it, right? So that's when we started developing CAS markers. At first, we tested 16 SNPs, and again, we had to face that challenge of dosage calling, right? So we were able to do it with the SNPs we got, and found, pretty high R squares for some of the SNPs we had selected, right? So these first ones are on chromosome 4, which is associated with melanogeny enterolobby resistance. So it's looking pretty nice. And also for the melanoma incognita, I just talked about, we also found one of these SNPs that can explain a lot of the variation here, right? We are currently applying it into breeding populations in Africa and in the US. So we are going to see how it's actually going to look like, right? Because we are just in the phase of validation at this point. Some other examples. So we got excited, right? So we were like, all right, so let's try it for other traits. Some of them were published, some of them are not published yet, which is the case of enterolobby, and also the case for sucrose, glucose, skin color, and beta amylase. But we knew it for beta carotene, so we were like designing Cas markers for these genes we found to be differentially expressed among the individuals, the parents of the population, right? So they would be our first targets for SNP designing. So we had designed 60 SNPs where 48 of them worked. So this is how the genotype calling, the dosage calling looks like, right? Some of them are very well behaved, some of them are not very well behaved, but that's the issue of working with hexaploids, right? Sometimes things get a little messier. But the good thing was that for the, so let me go back just very quickly. So you can see that some of these QTL are actually very close to each other on chromosome 3 and chromosome 12. These are actually in different places on chromosome 12, but the ones for sugar and beta carotene are actually pretty close. So we have other two on chromosome 12 in two different locations, and the beta amylase gene is on chromosome 13, right? So we have here all of them together. So these first ones are on chromosome 3, and we found a major SNP explaining the variation here for flesh color first, then skin color on chromosome 12. We also found one explaining almost 50% of the variation, and firmness, right, which is supposed to be related with beta amylase. But turns out the correlation between beta amylase content and firmness is about minus 0.4. So it's not complete, right? So we weren't expecting that high of a R square, but it seems that there is something that we can use here as well. All right. Something else we also tried to develop was Cas markers for quality assessment, quality control. This was actually the way we started, right? We had these from Dorcas, actually, right? She had designed some of these SNPs. So we tested first 30, and then we developed other 30 SNPs. Some of them worked better than others, but it turns out 34 worked at the end, and they are actually available on Excellence in Breeding website. So the list is there, and we hope to update them with the Cas markers for associated with traits soon. We soon realized that these, with the validation plates, right, that these Cas markers were actually very useful for, estimating genetic distances for distance and clustering analysis, trying to recognize some patterns in terms of similar individuals or different individuals or individuals from the same progeny and so on and so forth. But getting into the resolution of pedigree confirmation was not something that we were able to achieve with 30, 40 markers we had developed. We ran some simulations, right? And having up to 10% true assignments for crosses would require 90 markers without any missing data, which is very hard to get, right? And these for Um, and this is, of course, related with the complications of segregation in polyploid populations and species, and we are dealing with hexaploids, which makes things a little bit more complicated. So we are not so sure if Cas markers are well fitted for this type of work yet. Well, at least from the evidence, it does not seem to be enough, right? We would have to have at least 90 SNPs, 100 SNPs to be able to get those pedigree assigns, and then it would just get too expensive. We would just run mid-density platforms, right, instead. But interested in seeing, a little bit of diversity and how these diversity groups are distributed across different breeding programs. We ran a plate from Tari, Nakri, SIP Uganda, and SIP Mozambique, right, with 30 selected SNPs. Here are their dosage calling, the ones that we actually selected for analysis. One first thing we were interested in seeing compared to diploid dosage calling, right? Because one thing that people used to do is put together all those 5 heterozygous classes, right? And as we would expect from genetics, right, this causes lots of issues regarding separation of individuals, right? Because that means we are missing information. So some individuals which were clustered with diploid genotypes were actually in different, they're of course related, but they are different individuals, right? Some of them are still collapsed, right, into the same clusters. For some of them, we actually expect, because they're just copies or clones, right, in different locations. Like it has the same, like Tari has the same clone as Nakri, and they provide the DNA for it. So this was something nice to see also. The ability, right, to separate those individuals that it was giving us the same, based on the same, and this is again the same number of markers, right? We only increased the information by calling the dosage of these names, right, of the alleles. It also worked nicely for disambiguation. Again, some of the individuals were duplicates, right, across different, which were expected to be the same individuals, or actually not, right? They were placed in different places in the dendrogram. So this is actually something we need to pay attention in terms of naming those clones, things that do not relate with the actual genotype, right? And we were also able to find 3 major discrete ancestral groups in these 4 core collections, right, from each one of these breeding programs. They for Tari and Nakri, but not so well distributed for SIP Uganda, which has more of the red ancestral group, and for SIP Mozambique, which has more of the blue group, right? So from going from validation to the actual application, we tested, we fingerprinted our whole genomic selection populations from Nakri and SIP Uganda. We are also able to detect some mislabeling, right? Some individuals we would expect to cluster together, but they're actually far away. So these are all things that we are trying to figure out. And this is very important because now we are going towards genomic selection applications, and we annotate the pedigree, believing that that pollen came from that individual, right? So the name says one thing, but the genotype actually says another. And this, of course, has an impact if we are trying to use pedigree, for example, for prediction model, right? And we also wanted to acknowledge the idea of having the genotypes, right? Whole genome SNPs for figuring out these genomic relationships, right? But we are not there yet. We are still getting whole genome SNP data. So this is not something I'm going to be able to show to you here today. But just as final remarks, our goal here is to effectively apply QA/QC markers and investigate parental diversity, mislabeling rates, and things like that. Also incorporate marker-assisted selection for traits of interest, because I'm not sure if you noticed, but phenotyping nematodes, investigating some of those traits like beta amylase is not an easy task, right? It needs like several steps of experimentation, and using marker-assisted selection for such high heritable QTL would be something that would increase our genetic gains, right, for speeding up the process. Now we are about to release a chromosome level haplotype resolved genome for sweet potato. So that's for sure going to help us with marker development for these traits, because at first it was more like try and error, right? Now we can actually target the haplotype we want. We are also about to apply genomic selection models for early parental selection after testing, those models considering diploidized dosage haplotype markers. And we are also helping with the incorporation of mixed modeling, like using pedigree and spatial trends information, selection index into go into much detail on the methods we actually use, the algorithms we actually use for linkage mapping, dosage calling, and QTL analysis. But we are proud to be part of the Tools for Polyploids initiative. And we've been there for a couple of workshops, so they have all on YouTube. So if you're interested, the details, right? Thank you all for listening. And these are the people responsible for the populations and data sets and the analysis and at NCSU and EIB. You all.
Great. Guy, you are the first person in a while that actually keeps the time so accurately. Like 25 minutes, yeah.
That's what being a professor means,
exactly.
Right? Have students running out of your class, so you have to finish in time.
Exactly. Yes. I can see that you have all this, measured with the chronometer. Good. No, thanks, Guy. This looks terrific. I'm sure that there is several questions, especially technical. First, maybe I will just, read the questions that are in the chat, which are more, clarifying some concepts.
Right.
The first one, Bodo is just asking if sweet potato is an auto or an allo hexaploid consisting of tetraploid plus a diploid ancestor, or how exactly the genome structure of sweet potato, is conformed.
Right. That's an awesome question, actually, because we also asked ourselves when we started doing this work, and there is for sure this theory that it was originated from tetraploid and then a diploid ancestor. But if that was the case, they for sure have like very similar genomes because they behave as an auto hexaploid, sorry. Like their behavior in terms of genetics, recombination, even that this type of distribution here, it's typical from auto hexaploid species, right? If it was allo, we would see more things like this, right? They looking like diploids instead. So we are more confident that it's actually an auto, or at least behaves genetically as an auto hexaploid instead of an allo hexaploid.
Okay, interesting. I keep clarifying the concept. So Guy, just for everyone, when you say the pseudo diploid calls, you mean grouping all the possible heterozygotes as just a single class, right?
Exactly. So in this case means we put together all these individuals as class 1, these individuals as class 2, and these individuals as class 0. So as you can see, and any person doing genetic analysis would notice that it doesn't make much sense in terms of what we want to do, right? Because these individuals calling, right? That means that they all are placed together in the same cluster. So this is something that we don't want to, right? And that's why we are so interested in these a measure of quantities, right, of amounts of each one of these alleles, so we can actually call the dosage.
This is just another, clarifying question. This is actually from my side. Can you explain again what is this diploidized dosage haplotype?
Oh, yeah, sure. This is actually, I noticed that there is something missing here because it was supposed to be a versus among them. This is diploidized versus dosage versus haplotypes, right? Because if the population has a structure like a biparental population or multiple biparental populations, we can try to estimate haplotypes or microhaplotypes, right? So we increase the information. We go from biallelic from, going from AFLP markers, like dominant markers, to microsatellites, right? We increase our information because we go from not knowing, not recognizing the heterozygotes to recognizing the heterozygotes, right? So dosage is usually better than diploids, but it seems that for some traits, diploidized calls would be okay, but for some dosage is better, and for some haplotypes is better. But of course, our observations, and that's one of the reasons why I didn't bring it here today, are limited to to biparental populations. All right, so we do not have that evidence for more diverse actual breeding populations. That's something we are trying to get right now.
Okay. Yeah, then this fourth idea, should it say diploidized versus dosage haplotypes?
Exactly.
Yeah, okay. Yeah, thanks for that. I just got confused. I didn't know if that was a new concept that I didn't know diploidized dosage haplotypes. Good, thanks. Please, if there is any questions and comments and thoughts, please, this is the time to jump in. Gary Atlin is asking on the chat, all Cas markers are diallelic, I suppose, but alleles don't have to be. What proportion of, say, structural genes in sweet potato are multi-allelic? Even in biparental populations, some loci will have more than 2 alleles. Yeah, Guy, is there any, the idea of how, in general, if you were looking at a regular allele, how much we're missing by just only using biallelic markers? Given that I guess there is many possible alleles, or just by, having all completely covering the genome with the biallelic markers, we are able to recover all these
right
multi-allelic.
Thank you for the question, because this is something we also ask ourselves all the time, right? Because the limitation that the biallelic SNPs offer us is not the limitation that the biology of sweet potato actually offers us, right? Because it's a high multi-allelic, right? They are not limited by biallelic SNPs, and that's actually the whole concept we use for QTL mapping, right? So in this case here, we would have something like that. As increasing the root knot counts, these ones as increasing or decreasing the root knot counts, and this one being neutral in comparison to the population average, right? So only here we would have like 3, 4 if we count the ones from Beauregard also, segregating the population. So this is totally for sure. But in terms of using it for market-assisted selection, we wanted to approach in a more simplified way, right? One way of doing so is, for example, by looking at the comparison of individuals typed as 1 or 2 in this population specifically, and how they compare when they are mixed, right? And it seems that this is actually a dominant marker, so a dominant QTL. So you only need one of these haplotypes to be resistant, right? So knowing that, we knew, well, if we found SNPs that are associated, right, or in linkage disequilibrium with these alleles here of the genes, right, of the QTL, we are going to be able to capture that resistance, right? And that's exactly what we are doing now, and it seems to be working. But yeah, we always keep in mind, that idea that there are actually many alleles segregating in the population. We are talking about biparental populations here, right? So can you imagine what happens in a breeding population? It's impossible to think biallelic, right? And this is one of the reasons why we want to pursue the haplotype idea of genomic selection, because it might not be or make much of a difference for diploids, which actually does, right? We know that now, but for auto hexaploid individuals, heterozygous populations, we know that this is going to be important for sure. We just need to deal with the complications, right? And having the right method, having the right data to be able to build such a tool.
Great. Thanks, Guy. That was a good answer. I'm gonna go, to Wolfgang. I think he has a question or a comment. Wolfgang, please go ahead. Just keep in mind the time.
Yes,
Thanks.
yes, of course. Uh, Perhaps I have here some background noise. If it's too loud, let me know. It is so, it goes directly into this direction. I think we must, I think some things which we observe in sweet potato, that a B-allelic model is simplifying the reality extremely. As we must think about what happens when we have a strong multi-allelic effects. Also, let me give one example. I think one thing which can't be explained is that with few introductions, a huge amount of genetic diversity can develop in sweet potato in a new region or area. A classical example is a secondary diversity center around Papua New Guinea and the Irian Jaya, the Indonesian part. I think it's estimated that only 15 to 20 clones arrived in this region, and then already 50 to 100 years later, it was the secondary diversity center of sweet potato. And I think such things in sweet potato, which you can observe, can only explained with multi-alleles carried by one genotype or a few genotypes. This might also be linked with the situation that, this is a hypothesis from my part, which I will never verify during my career. But this is, I think, the genetic diversity, what we observe, or the genetic variation, what we observe in breeding populations is going up, it is not declining. This was my comment. Either we must keep this in mind. I think BLA-like models work very well in diploids, but perhaps we do harm if we assume this in polyploids. Although this is what I have, although this is something for the next generation to think about. It's not my generation anymore.
Thanks for sharing, Wolfgang. Those are good comments. Something to keep in mind, especially for everyone working in polyploids. Guy,
And it's been a long road, right, Wolfgang, trying to make people aware of up in terms of knowledge and information in terms of markers, right, for a sweet potato. I think we are getting there. Thank you.
Nice, thanks. Guy, there's still 2 questions in the chat. Well, 3 actually. The first one is if you could share some ideas or thoughts on whether inbreeding depression in sweet potato is best explained by a 2-allele or a multi-allelic model. I'm not sure if maybe you understand better that question. Yep.
Yeah, I think it's a good question, but we are not there yet to answer it because we know that there are self-incompatibility going on in sweet potato, but we are not so sure about how many loci are involved with it, and if that plays a major role in terms of inbreeding depression. I believe it should, but we do not know much about it, so it's hard to tell. But out of the books, and the things that people are using to try to address that issue, it's usually based on two-allele model, right? But I believe it's limited, but we need to expand that idea. Unfortunately, we do not have an answer for that yet.
Interesting. Related question, Wolfgang, I think you want to give a comment on that.
Yes, exactly, because I want to share you with something where I was wrong. And this was at the beginning when I started here at CIP. There were a budget when the position was vacant for 1 year, and the assistant at that time made something he was interested. And he were using some clones which could be selved. And then it was so it was not a complete diallele with these 8 clones, but he wanted to determine the effect of inbreeding. And at that time, when he talked me about this, told me about this, I mentioned, this effect can't be so large. This is the inbreeding coefficient in the BLA-like model due to what in this famous book of Gallet about quantitative genetics of polyploids. And I said, no, the effect will not be large because the inbreeding coefficient goes so slowly down. And then come with me. And we were going to the fields, and the performance of the self-offspring were really drastically lower compared to the crossed clones. And there I was really, and at first I thought perhaps they have done something wrong here. So this is an experiment which is now should be made now for in the next generation. What happens? What do we observe? Is theory with a BLA-like model consistent with what we observe. I think this would be very interesting.
Yeah, it is indeed. I assume that some of the new seed breeders in sweet potato are going to try this soon. Let me just move
Also,
to
Gui would be, I think, very interested in such an experiment.
Yep,
that would be
for sure.
good. I will move to the next three questions because there's more and more in the chat. First, Guy, there is another question on whether the G blob works similar in diploids and polyploids. Specifically, is the GRM calculated in the same way?
We make a small modification, yes, but it's doable. It's something we can use for hexaploids individual populations for sure. GBLUP and the genomic relationship matrix calculated more or less in the same way, but with some modification because of the plot level, right? But it's doable.
So if I'm not wrong, it's just the allele dosage matrix from 1 to 6, I guess, in this case, but the denominator would change a little bit, right? Because of the frequencies.
Exactly.
There is another question, a bit more technical. They are asking, many thanks for the presentation, Guy. Dosage calling is performed at population instead of a sample level. In this sense, the read counting for each sample must be similar to ensure correct clustering of genotype calls. Is it possible to perform the dosage calling if you have different read depth among your population samples?
Right, like from the data, at least for genotype by sequencing data, right, we have observed there is actually this variation, and we can call the dosage, right? We perform some filtering. I think it's like for this population, it must be around 100 reads minimum, right? So that means we would need, or 50, I don't recall. But we would need to throw those individuals with very low read depth out, right? We filter it out, and then we perform the analysis. Having them at the same level of depth would be ideal, right? But it's something very, very hard to get. It seems that's something we can get from CAS markers, right? At least for most of them, we can actually have them at the same level and call the dosages well. But yeah, for genotype by sequencing data, it's hard, it's tricky.
Yep.
But
so
it's like,
naturally, you will find a lot of variation then on the read depth.
right.
So I guess the answer for for this person would be that it is possible. That's indeed what you would do in GBS.
One way, and people are trying this right now, would be to consider, the dependency among marker because of linkage, right? At the same time as you call the dosage. So maybe one wasn't in a really high depth, but then the other was, so you can kind of balance that, right? So that would help us with getting more SNPs. But it's something that we are still trying, actually, right? It's an idea.
Great, thanks. There's a comment from Marley. Marley, do you want to maybe, just give the comment out loud? And because I know that this is something that you're particularly good at, about the increased breeding depression. I guess is related to the comment from Wolfgang. Is that correct, Marley?
Yeah, no, thanks, Guy, for the discussion, and thanks for everyone for throwing comments. That data set sounds really interesting, Wolfgang. I just wanted to throw out there that that pattern of inbreeding depression you see doesn't necessarily have to be due to there being multiple loci, multiple alleles per locus. It's just sort of something that arises by how much inbreeding you get in the sweet potato. Because it has more alleles, because of the way it inbreeds, you would actually expect more inbreeding depression when it does get inbred. But I don't really want to go into the details because that's not the focus of the talk.
Would it be a good experiment? Yeah, sorry. Go ahead, Marty.
While I'm here, I would maybe throw a question to Guy, just since we have a few minutes. Yeah, Guy, thanks. This has been great to see. I know a lot of your work, but some of this that's been a little earlier, I maybe have forgotten. But would you have any like general reflections over, sweet gains and moving to SIP and everything, about what it's taken to move sweet potato into being a genomically bred crop, and really what it took to build all that?
Right. I think it took us, thank you for the questions and the comments, Marley, and for all the help for this work, actually. Yeah, that's for sure. I think it took us a long time, like to face the challenges of getting, sweet potato sequenced and genotype by sequenced and things like that. And of course, that has an implication of us getting right into genomics assisted breeding, right? I guess EIB has helping us a lot with, the operational aspects of it. Which is, of course, very important and sometimes the most limiting part. But the tools and the methods are still being developed, right? And even the populations and the way the breeders build their populations is different, right? So we have polycrosses, we have biparental crosses. Some of them are trying to work with hybrid breeding, right? So in addition to the complications associated with the genome itself, right, we still have these many different breeding schemes, right, which is working as we know now, but we are not so sure how we are going for in the next steps, right? I guess having the whole genome markers and running some, proof of concept work for genomic selection is going to help us to understand which approaches would work better for sweet potato. But at this point, it's hard to grasp. So I understand that most of you guys are working with other species which are long inserted into genomics assisted breeding, right? But I guess that's the issues we have, right, with working with polyploid species and hexaploid species in this case. But yeah, we'll see. We are excited. We are happy to see the Cas markers working and people are applying it into their breeding program. So we understand this is an advance, but we are still missing the mid-density platform for sweet potato. So we have it to do it in-house, right? Which actually takes a little bit longer, but I know Breeding Insight and EIB are working on the DarkTech platform for sweet potato. So we are looking forward to test it, actually. I think this is gonna be a game changer for us, for sure.
Thanks, Guy. Yeah, I'm really impressed to see the trajectory of all this work. So thanks for your presentation.
Awesome, thank you.
Great. Well, once again, thanks, Guy. That was a terrific presentation, and thanks everyone, for the questions and comments. Some people already started to leave, so I think, Guy, this is the right time to finish the meeting. Not without really thanking you once again for taking the time to share all this knowledge, tools, methods, etc. I think everyone really appreciates it, and I'll make the recording available because there were several people sending messages that they unfortunately had another meeting, but they really wanted to see at least the recording. Thanks again, Guy, and thanks everyone. I wish you a great day. And if you have any other comments, please just drop a message to either us or Guy if something was not addressed. Thanks everyone. Have a great day.
Thank you, you too. Bye.
Thanks,
Thank you.
Guy. Bye. See you.
Thank you, everyone.
Thanks, everyone. See you.
Thank you, everyone. Eduardo.
Was that Skiru?
Yes.
Okay. How are you?
How you doing? I'm fine, and you?
Yeah, pretty good. Just starting the day. Good way to start the day with a good presentation.
Okay. See you.
Yeah, see you. Take care, everyone. Stay safe. Bye-bye.